Okay, great. Hi, everybody. Welcome to the next talk by Alexandra Born. And I'm kind of excited for this talk. So actually, the first time I met Alex was a couple of years ago at the Rocky Mountain Conference in Denver. And I think we met at a coffee shop somewhere. And so with Nushi, she was proposing back then her project. And it was already interesting for me because the idea was to use EPR restraints to validate structure and domain confirmation by EPR restraints, and then so utilizing EPR as a validation tool. Now, the really amazing thing is, and I think most of you guys will agree, EPR is great. DEAR is great, but it's really powerful if you actually combine it together with other biophysical methods, such as, well, here you go, NMR, solid state NMR, liquid state NMR, cryo-EM, you name it. And I hope all of you guys will become convinced by this talk that that's just the way to go, and we can do future application with it to eluding biomolecular question and gaining answers to it. Now, without further ado, I think, Alex, I'm going to give you the show, and please go ahead and enjoy the talk.
Thank you, Thomas, for the introduction. Let me share my screen. Let's get my laser pointer. Okay, can you see?
Yes, I can see very well.
Let me do one thing really fast. Sorry. Okay, let's. Cool. Okay, yeah. Well, thank you, Thomas, for the introduction. And thank you for inviting me to speak as well. And like Thomas said, I will be talking about using DEAR distance distributions to cross-validate multi-state NMR ensembles. So first of all, a little bit about me. I'm currently a postdoc at UCSF in the labs of Ashish Manglik and John Gross. But today I'll be talking about my PhD work while I was in the lab of Beat Burgely at the University of Colorado, Anschutz. And I am without a doubt an NMR spectroscopist and not an EPR spectroscopist. I'm sure you all know my friends over at ETH Zurich in Gunnar's lab, who did all the measurements of this project. And so if you have any questions about any technical aspects of EPR, I would definitely urge you to reach out to either Gunnar or probably Janne at this point. Would also be able to let you know anything more specific about the project, because they more or less just gave me the DEAR distance distributions and I just used them. They know all the ins and outs of the measurements. So yeah, so our project has a couple different, is very, very much multifaceted, but one of the things we're very interested in is like domain motion. And this is a really interesting thing to look at because, as I'll get to the next slide, like a lot of proteins do have multiple domains. Another exciting thing about structural biology right now that people are excited about, but also kind of afraid of, is like this AlphaFold that came out recently, and that's the fact that it's gotten really good to predict structure. But there's actually quite a few things that AlphaFold is not very good at. So like things like multi-chain complexes and like IDRs, proteins with multiple conformations, which I'll kind of talk about later, are actually really important because, as we know, most proteins are not just a rock. Most of them have dynamics. Another thing AlphaFold is still not great on, but they're okay at, are like the relative position of domains. There are definitely multi-domain proteins that do like pack together, but there are also other proteins that have more flexible structure in between those two domains. The protein I'll be talking about, PIN1, I'm showing you the AlphaFold model with the two domains in orange and blue. You see here, it's kind of in this compact state where they're interacting. And this is like the most recent model from the most recent edition of AlphaFold. There's another program that's more supposedly better suited for like multi-domain proteins called Demo. It's like a version of ITASR, but you see in this model that the two domains are very much this extended conformation. So I kind of just wanted to highlight that this is like not a solved problem. Like these prediction programs are still not great and have not converged on like figuring out the exact position of the domains. And that's why I think EPR and NMR are really useful. So just giving you a little bit more background about multi-domain proteins, about 80% of all eukaryotic proteins have multiple domains, yet less than 35% of proteins deposited in the PDB contain multiple domains. So I think overall, they're kind of like understudied and they're hard to represent and study structurally. Something else that's kind of interesting and really interesting about them is there is this domain motion that can occur and that can, and this leads to some structural and functional flexibility. Like for here, this is a simple example where you kind of see this elongated state with this 2-domain protein, yet upon binding to this DNA that you get this kind of more compact state. Also, where two proteins interact, that could lead to potential regulatory or allosteric sites, which we kind of talk about a little bit for my project as well. But you can get overall for multi-domain proteins, you get these large domain motions that are interesting. And overall, it's really important to study the whole system and not domains in isolation. I think it's a really common theme when to study a protein that has multiple domains to cut it up and to study the domains in isolation. But by doing that, you get rid of the domain motion, you get rid of these regulatory and allosteric sites. So you're not really studying the protein in its natural environment, an environment being in condition of the other domain nearby. So overall, like while it is useful to study in isolation, it's also good to look at it for the whole system, to study structure, orientation, dynamics. I think these three things, you can do all these three things with NMR. I also think EPR is really useful, as well, as you'll see later on, to get at these parameters. So again, this is probably something you all know, but I'm not an EPR spectroscopist, but I think EPR is great, especially DEER, for like figuring out domain distributions and like domain motion. So I'm just kind of picked the two simple examples of a 2-domain protein. If it's kind of static in these two domains, you kind of expect just a single population at that distance between the two spin labels, versus if the two domains are pretty dynamic, one relative to the other, you kind of get a more dispersed distribution. But then there are systems also that can have both, which I think are kind of interesting, where you can have some instances of things being this compact state and some with this extended state. And while these pictures are kind of nice here to give you kind of an idea of the story, it is still pretty simplistic. And when you I think another theme of my talk is how intra-domain structure impacts inter-domain distance. So while it might be easy to think how the intra-domain structures of these two domains are different than these two, and that's why this one is more static versus this one's more flexible, I think it's more difficult to think of and conceptualize how a protein can be in both compact and extended states with like the same like protein structure. And that kind of gets into this like multiple, we solve multiple state ensembles to kind of solve this problem. And it's dependent on like motions of like side chains within the domains that are causing like larger changes between the two domains. So while EPR is great at giving you like these kind of like really nice reports on like the interdomain distance, I think we are also really interested in like what distinct conformations exist between these two states as well. And that's kind of where an NMR can come in and fill in the gaps. And that's why I think EPR and NMR are great to like utilize together. So as you probably get into, based on what I've been talking about, the protein I solve, you'll actually see distributions very similar to this. So the protein I'm working on, I worked on for my PhD, is called PIN1. It's a phosphorylation dependent peptidyl prolyl cis transisomerase. So it isomerizes prolines that are immediately preceded by either phosphorylated serine or a threonine. Biologically, it's interesting. It's like upregulated in cancer, downregulated in Alzheimer's, but overall, we're kind of working on it more because it's been heavily interrogated with NMR as like a model 2-domain protein. So yeah, 2-domain protein that has this flexible linker This is the original crystal structure that was solved, where the two domains actually interact in this inter-domain interface. And the two domains are connected via a flexible linker. The WW domain is here in orange. The actual catalytic domain is in blue. Both domains bind, but only one domain isomerizes. And there's a bunch of evidence suggesting that the WW domain regulates the activity of the PPIase. I'm not going to go into that much today, but just so you know a little bit more about the system. And like I mentioned, the original crystal structure is shown here, where their two domains are interacting through this interface. This is kind of what I'd show as a compact state that you'll see quite a bit of, and like I'll talk about what it used the word for. But also like an NMR structure came out a couple years later that did not contain any interdomain restraints. So this is just cycling through the different states of that NMR structure. You can kind of see it's this extended state. So the crystallographers thought it was compact. Some NMR spectroscopists first thought it might be extended. Further NMR research, like looking at tumbling times, actually showed that something in between and compact and extended actually existed. So both might be right. We just don't know the proportions of compact versus extended. And while this protein has been looked at, like the NMR structure was solved in 2003, there's only been only the last like 2 years of people have been able to put some type of populations of this compact and extended state. And then ours is using EPR, which is pretty sensitive. So overall, our working model is that this WW domain influences, like its structure influences the interdomain distance distributions that then impacts the PPIase structure and activity. Like I mentioned early on, my theme is that intradomain structure impacts interdomain distance and vice versa. So I think one of our other goals was to correlate interdomain distance to intradomain structure. I will mainly talk to you about, I won't be getting too much into the actual correlations. I'll show you a few, but we'll be mainly sticking to like what I did to validate the structures. But I'll show in a minute where you can find out more. But like I said, ApoPin1 is samples extended to compact states. We don't really know the ratio. There's also evidence suggesting these are both peptide ligands, that they specifically change the distribution. One has been shown to stabilize the extended state, while the other, there's some evidence of stabilizing the compact state, but other evidence that suggests it doesn't change. So that's something else interesting. I won't really go into these large changes of the structure, but I'll show you a little bit of work on that. So like overall, like what we want to know, like what are the populations and positions of the extended and compact states in ApoPin1, and like how do these ligands change the equilibrium? And then also briefly, I'll get on what correlations we make between interdomain distance and intradomain structure. Overall, how we did it was using NMR and EPR experiments, and we solved multi-state ensembles of ApoPin1 as well as these two ligand-bound forms. Again, I won't be going too much into the multi-state ensemble, but again, these are things that my boss Bayat has worked a lot on optimizing, and these ensembles show like dynamics in the system through structures, simply put. So again, today I'll be mostly talking about the populations and then also the positions of the extended and compact state, because while EPR kind of shows you the general position of the two spin labels, it's really hard to visualize without other methods. So most of what I'm talking about today can be found in our most recent publication in JACS, but also there's two other papers that are of relevance, one on residual dipolar couplings, and then another paper we just submitted this morning, actually on our ligands. But most of what I'm talking about is in this paper. So if you have anything specific and want to know more, I please urge you to also look there. So overall, what I'm talking about today is like using these Dieter distances and distributions to cross-validate NMR structures. So I kind of have a general pipeline of what I'll be talking about. for the remainder of my talk. We're starting down here first. I'll be talking about my NMR structure restraints. Generally how I solve ensemble. I then import that into this program called MMM that you guys may all know of, to simulate the Deer distribution of this ensemble structure. And then I also then have measured these experimentally and then compare them. So to start, I'll start with my NMR structure restraints that I've used for my structures. So we mainly use these 4 restraints to solve the structure of pin 1. You guys may know the NOE, but we may get more precise by the exact NOE, but this is the distance between 2 protons. We can get distances up to about 8 angstroms. These distances can be very precise, but again, because it's only up to 8 angstroms, it's really hard to measure any intradomain, especially when there's instances of an extended state in like in pin 1. We also use scalar couplings, which are just simply bond angles to orient our bonds and atoms. Residual dipolar couplings we also use, and these are angles that are relative to external magnetic field. And these actually do kind of start getting into some interdomain distance and relative position of one domain relative to the other. But I think most importantly, I can say most importantly for my ensemble, at least, was using paired magnetic relaxation enhancement. So these are distances between a proton and an electron, and we can measure from about 12 to about 24 angstroms. So these are a great way of getting at these longer distances between the 2 domains as well. And these can be used almost like NOEs into a structure calculation. You get an effective distance like you would with an NOE and use that as a restraint. So that's definitely probably one of the more important ones for our actual structure calculation. And then I'll talk about in a minute, but we didn't use Deer for actual restraint. We just used it for cross-validation. But as you guys probably all know, these are great at measuring distances and distributions up to about 80 angstroms. So a great way to actually look at interdomain distance between 2 electrons. I just want to show you some how much data went into these structures. I think we measured a ton of interdomain NOEs, over 2,000 on pin 1. They're drawn on gray in this structure here. We also were able to measure about 20 interdomain ENOEs, and you can see them right here. That does not disprove existence of extended state. It just proves that the compact state also exists because of this weird, not weird, but all NMR restraints are measured in solution, while dynamics are undergoing during your measurement time. So everything kind of gets time and population averaged. So it makes populations really hard to disentangle. Also, we measured a bunch of scalar couplings to also get more bond angles. These are the primary restraints for looking at the intradomain structure. But again, we have some interdomain ENOEs that also help position the 2 domains in the compact state. And then between the 2 domains, we use, again, PREs. Here I just have one of MTSL labeled and then various nuclei to proton distances, just to hopefully give you an idea of how these distances can orient one domain relative to the other. We had 4 different spin label positions, and like I mentioned, measured them with the MTSL spin label. We also someone else measured about 400 RDCs that we were luckily enough to be able to use for our structures. And like I mentioned previously, that all these NMR restraints are time and population averaged, unlike Deer restraints. So those are restraints that we used and measured on pin 1. Now I'm going to jump to the Deer measurements, just because I'm talking about actual experimental data. And like you all probably know, Deer is a great way to measure the distance between 2 electrons between 15 and 80 angstroms. Again, we use our same PRE mutants that we did for a single mutation and single labeling site to then do 2 of them. And then unlike NMR, we actually flash freeze these samples. So we can actually measure all the distances at time of freezing. And so we can get a population distribution. And then, like I mentioned, this was all measured by Gunnar's group using either 4 or 5 pulse DIR. Just to give you an idea, we measured 5 different combinations of these spin labels. Here are 2 that are within the same domain, so we kind of expected a little bit of a narrower distribution. These are both within the PPI-ACE, that larger blue domain. But more importantly, we measured the distances between the 2 domains, these 3 combinations here. I'll talk more about one of these in a minute, but we were able to use both model-free approaches as well as using, for these 2, we were able to fit them using a 2 Gauss approach, which I'm excited about, and I'll tell you why in a minute. But yeah, we were able to, but as you can kind of see already with these 2, that we see this short state here, as well as this evidence of a longer state as a longer extended state. So going into detail with one of these distributions, here, this state matches pretty well to what you'd expect in the crystal structure. And this short, narrow distance is kind of like what we expect to be the compact state. And then again, we have the evidence of this longer extended state as well that would match pretty well the original NMR structure. And I'll actually show you that in a little bit as well. But yeah, we were able to fit these using like 2 Gauss approach. So we were not only able to extract the distance and distribution, or deviation from that distance, and for both Gaussian equations, but also the population. So this, the data here shows that the compact state populates about 68% in pin 1. A similar distribution from a similar mutant or spin pair showed about like 73 or 74% population for the compact state, but which is in pretty good agreement with this other one. So we kind of say that it's about 70% compact and 30% extended. This will be the last time I really talk about the ligands, but when we add one of the ligands, it really doesn't change necessarily the population. When we add this one ligand, maybe increases a little bit, moves it up a little bit, makes the distance a little bit longer maybe, but nothing drastic. Versus when we add this other ligand, CDC25C, we see the compact population drop, the extended state population increase, which is kind of what we expected and what has other evidence has shown as well. But again, no one has really been able to put a population on it until now. So for the compact state, we say it looks pretty much like the crystal structure. I'm okay with saying that. We'll see in my NMR structure that's similar as well. But we really don't know the actual positions of the extended state. Like the original NMR structure kind of showed the position in various states, but we don't actually know what fits well under this curve. That's kind of what we'll do with our NMR ensembles shortly. So moving on to actually what our NMR structures look like and how we calculated them. I'll kind of go you through a general Siena approach using those NMR restraints. So like I mentioned, we use these 4 restraints in our NMR structural calculations, and again, not using the DIR distances. So because of how we use this program called Siena, written by Peter Guntart, also in Switzerland. And I won't go into heavy detail about why we had to do this, but because we had a multi-state ensemble that we needed to not force the 2 domains to always be close to one another. And there's this parameter that makes you keep, when you do a 2-state structure, like those 2 states have to be put together when you solve it. Because of this, we had to kind of go about a 2-step approach that we basically had to solve the structure of the WW domain and then separately like solve the structure of the PPI-ACE domain, locking in the angles set from step 1 in the WW domain. So like I showed you here in the first step, we just use the intradomain WW restraints. The WW domain is here is shown kind of overlaid. PPI-ACE is kind of wherever it needs to be for this first step. It's not that important. And then for the second step, we actually use all restraints and all those intradomain PREs, as well as our RDCs, to then orient one domain relative to the other. So here I'm showing you the PPI-ACE domain overlaid, and you can see the WW domain. And this, I forced this program is in a, this calculation was a 2-state structure. So we see one position here in our typical compact state, then another position in this extended state. But we can allow multiple different structures and different number of states for this as well. But in general, that's like the procedure we use to solve the structure. And then we now can, with our various structure calculations, we can then simulate the DEAR distributions via MMM. So multi-scale modeling of macromolecules was, again, with Gunnar's group. What's interesting and what's kind of great about this program that I like using is, for one, it's really easy to use. And you can simulate this Rotamer library. So we were able to simulate our MTSL positions of our 4 spin labeled sites. The centers are visualized by spheres that are proportional to the population. So you can see some regions like the spin label is pretty dispersed versus like another region, it's more centered in one position. And then based off those Rotamer distributions, you can then simulate a DEAR distance distribution between those 2 spin pairs. So for here, this example is looking at the original crystal structure where you only see that compact state. And I'm showing here the 3 different interdomain distances. You can see those 3 positions that they measure. That's all pretty like relatively narrow. The variation is actually more due to the Rotamer spin's position rather than the protein itself being flexible. But yeah, we can do that and we can compare it with our actual experimental data now. So experimental is in blue. And as you can see, this is our measurement on ApoPin1 using a model-free approach. You see that our compact state, the crystal structure of one pin overlays well with this large, what I can term the compact state, which is kind of what we expected. But again, we still have this extended state that is not able to be fulfilled with this crystal structure. And versus if we looked at, instead of looking at the crystal structure, we looked at the original NMR structure, which didn't have any intradomain restraints. You actually just see this extended state. And again, the simulated is in red here, the experimental is in blue. You kind of see that these really don't match well at all either. You see instances of this extended state, but really nothing of this compact state. But nonetheless, this approach is great for like looking at various NMR structures and comparing it to your experimental data. So lastly, I'm actually going to show you like our final structure. But I use this model to kind of look at a couple different parameters that we had to optimize before we got our final structure. So I listed 3 of them here that we looked at, but the one I'll talk about is the number of states in the ensemble that are necessary to fulfill these DEAR distributions. So I guess, so when you typically solve a NMR structure, most people will solve it as a single state and try to use all the data at once to get just one average structure. But that doesn't necessarily satisfy all the data well. Often because of this averaging that occurs during your measurement, things just get extremely convoluted. And if 2 states exist within the same measurement, like we have in my case, things get skewed. So it's really useful to use these programs where basically the program like Cyana has 2 chances to fulfill all the data if you're using like a 2-state approach. So I'll show you in a minute, but like often when you add more states in to be used, your restraints can be used better. So here I'm just showing you like if we solve a single state structure of pin 1. Again, here I'm just showing you, these are like the crystal structures and the distances I'm showing in these distributions. You can even see that compared to my single state structure, that orange domain right here does not match the original crystal structure very well. And you can even see that their simulated ensemble in red does not match the experimental very well. But when we allow 2 states to fulfill all the NMR restraints, this is kind of the structure I showed you earlier, where you have the one position of this orange domain here and the other one back here. And now you can see that these, the ensemble simulated distribution actually matches pretty well to the experimental. And What's not perfect are the populations. Like I mentioned earlier, NMR ensembles are really not sensitive to populations, but more to like positions, versus deer is more sensitive to populations. Because I told this program to run a 2-state structure calculation, it'll have these 2 states in 50-50 population. But we know from our actual deer experimental data and the Gaussian fits, it's more like a 70% compact and 30% extended. But yeah, so our 2-state structure, I think, actually matches the experimental deer data extremely well. But adding a 3rd state really does not improve the model at all. You see that there's kind of an additional state that's kind of added in between the two, but no new populations are getting added that help satisfy the experimental data better. So this kind of made us for like why we kind of have gone with like looking at a 2-state structure of pin 1. Often when you solve a structure using this program called Cyana, when you choose a multi-state model, you look at this thing called target function to actually help decide the number of states that are necessary. Often you'll like choose the minimal number of states that fulfill all the NMR restraints, but the target function will drop almost for every single state you are adding. But you don't want to overfit. So you kind of choose the minimal number of states that fulfill all the data pretty well. So for this regard, 2 to 3 states also fits the data pretty well, all the NMR restraints well. So that's kind of why we're kind of happy with our 2-state model. But we are definitely picked the 2-state model more because we got like extremely good fits compared to the experimental data. So just to give you a little bit more, looking at the structure. So this is our 2-state structure of pin 1. You see this compact state and this extended state. Again, here's just like a blow up of the deer distribution, where compact state is this short population versus this this other conformation that has actually never been shown to exist, actually fits pretty well to the experimental data. It's more dispersed than the compact state based off of this, which is what you expect. Because in pin 1, like while there is relatively like one compact state, there are a couple different positions of extended state, but generally, at least, it's in this position around the back side. But again, it's not just one position like you'd see with the compact state. And then again, like I mentioned, based off of our experimental deer data, we can say that this population is what's occupied about 70% of the time, and this one's about 30%. So last thing I want to talk about briefly is I kind of want to talk, a big part of my project was also looking at these correlations between intradomain distance and intradomain structure. So we solved this 2-state structure, and we can then look within each domain to look at any correlations that exist. So we basically to just compare the structures of the compact state versus the extended state. And that's what I have shown over here, with the extended state in this teal color and then the compact state in the darker blue. I just wanted to highlight a few regions that we see changes. This position right here. So overall, we're looking at the PPIACE domain, which is that larger blue domain. In the compact state, this is where the WW domain is like interacting with, like right here. So it's kind of not surprising that we see a change in the interface between the compact and extended state, because like the presence of that WW domain, whether it's there or not, could change that region of the structure. Like I kind of mentioned and briefly alluded to, that there's evidence of like some allostery that's occurring in the system. So the idea is that the presence of the WW domain, whether or not it's here or in the compact state or somewhere else in the extended state, could then therefore change the structure somewhere else in the protein. So we also see change in the structure on this backside here, and this is where the active site is. And you see some instances of some small changes as well. And so these changes are seen in just like looking at the apo structure of pin 1, again, not with any ligands, but we later will see that ligands also change the structure even more, and also change some of these correlations as well. So overall, today I showed how we use experimental deer measurements to cross-validate these interdomain positions of these multi-state NMR ensembles. We showed that these, we untangled these extended and compact states in pin 1. We saw that they pin 1 occupies compact state about 70% of the time. We showed there's this novel, this extended state often occupies this position around back. We also saw some correlations between intradomain structure and interdomain distance. Some future directions I know the lab has in mind are also to actually use those 2 Gauss equations as structural restraints rather than just using them for cross-validation. But I think overall the bigger picture was hopefully to be able to use this similar model to like work on other interesting multi-domain systems. With that, I'd like to thank my old lab, and then especially Beat and Marcus, and then Gunnar's lab, especially Janne and Frauke. Lots of people working on this project. I was also really lucky to be able to attend this EPR summer school, I guess 2 years ago, a little over 2 years ago now. That was actually in Denver, so I was extremely lucky to be able to attend. And then I met Thomas also at that conference the week after, but also like to thank you to the EPR Society for inviting me to speak. So with that, I'll take any questions.
Thank you, Alex, for the amazing talk. I really appreciate it. I mean, it really shows how powerful combining 2 biophysical methods can be.
Yeah.
So I'm just gonna start asking people if they have a question. If you do have a question, just interrupt, just unmute yourself and go ahead. In the meantime, I'm just gonna ask maybe 2 questions. One time was, so you said you adjusted the weights for the PRE. I mean, the PRE will obviously give you some information on the exchange from the 2 domains. In addition to it, it's not too bad in giving you population once you have the exchange down. Now, when you said like adjusted the PRE weights, did you refer to that you looked at the exchange rate, or what do you mean by adjusted the PRE weights?
Yeah, that's very fair. I did not go into very much detail with that because we didn't just use PRE as like we actually can like measure the difference between the rates, like the relaxation rates, to actually extract a distance. Then we use that distance itself in the structural calculation, kind of like an NOE. So versus like other people will just use PRE as like more like, you know, this is the number, this is the relaxation rate, and then that, like, I think NIH Explorer is good at using that in a different type of way than like Cyana, versus Cyana used it as but that's the thing, the PRE, so the weight itself was more like the weight of that distance restraint relative to like the other NOEs.
Yeah, makes a lot of sense. Yeah, okay. I didn't know about Cyana, how it handles that, but yes, you're right. In Explorer would be the rate, and then you would get an ensemble calculation based on that. Now in Cyana, I don't know. Another question was, how did my friend Nick Sarakis get on the paper? I'm curious.
So he actually measured the RDCs when he was with AD.
Oh, son of a gun.
So it was really funny. So I met him I was giving a poster presentation at ENC in 2019. He came up and like told me, and he and Bea, I think either like overlapped by like a month or like not at all. But regardless, they never published a paper or used them for anything. So he just sent them to us. And then we had to figure out what to do with them. And it was not trivial at all. But yeah, that's the story. I mean, it's also like one of the great things about like why you should go to conferences, because like I met you and I met him. I would have not gotten that data if he hadn't come up to me. So
Justin.
Hi, Alex. Very nice talk. I had one question about the very end. When you were from your distributions, you saw evidence of multiple states in the active site. I was wondering if you tried to validate that by going back and looking at the HSQC, let's say, and seeing if you see multiple peaks, or if you see evidence of any kind of conformational exchange.
Yeah, so interesting. We see like chemical shifts, or no, not necessarily. It's kind of hard. We do see some minor like changes in dynamics, but it's not on like a slow exchange regime, so we don't see multiple peaks. They're mostly just like little blips in like R1 row, and that's kind of it. Any other questions?
Well, while we're waiting, I might as well just ask a couple more questions.
Mhm.
Now, in my experience, I mean, that's more an EPR question. So in my experience, like I suppose how the EPR was measured, the Dion specifically was glycerol, right? So you have added glycerol and then you slow freeze it in liquid nitrogen.
Mhm.
And then you perform a measurement. Now, did it ever come to you that maybe the glycerol has some effect on the populations? And the reason why
Yes.
I'm asking is it should be mirrored in the PREs eventually, because
Yeah. It's a really good point. I actually did not measure PREs with glycerol, and that's actually a really good idea.
no, you can't. I mean, this is impossible to do.
Oh, okay. But yeah, so we did think this through, and one point, like Frauke, I think, told me like, oh, she's not too worried about it. But I actually did notice this in another thing. So while I didn't see slow exchange peaks in the active site, I actually saw some slow exchange peaks in the interdomain interface. And we actually did see changes in, we actually did see in the spin labels itself, like while we did validate that the protein was still active with the spin labels, we saw those populations in the interface actually changed a little bit.
Yeah.
And they actually, like the populations we get out of that region actually match the populations pretty well. I mean, not great, but like okay compared to the deer data. Regardless, we're not that worried that glycerol itself was changing the distributions. What was more likely to change the distributions was actually the like spin labels. I can that's kind of coming out in the next paper too, so I'll let you know.
Okay, so I suppose
like-
you guys used the gadolinium label afterwards to look at PREs, or what did you guys do?
Say that again.
You said that in the next paper, you.
Oh no, I just with like the ligands. When we
okay.
actually worked with the ligands, we kind of get more into that as well. Like looking at these peaks in the interface.
Yeah.
I just don't have that pulled up to show today. But again, we weren't looking at glycerol specifically, but we have like good evidence suggesting that the glycerol itself wasn't changing it, but other things were.
Right.
So a little, but it's only like a little bit. It wasn't like reversing like the it wasn't reversing the populations at least.
Right.
Like if anything, we thought it would, stabilize the compact state is what I would at least assume. But we actually don't see that necessarily.